Scripps Research Fully Interactive Resources for Students and Teachers, or FIRST. My name is Rosie Alvaran-Seckler, and I will be your moderator this afternoon. If you're joining us for the first time or you're just not familiar with us, Scripps Research is one of the largest nonprofit private biomedical research institutes in the nation, and our mission is to develop the next generation of medicines and diagnostics to address the most present health problems in the world, such as COVID-19. I want to encourage all of you to visit our website after the webinar, and if you have any questions, please contact us. We hope you enjoy today's webinar. For today's presentation, we'll have graduate student Melissa Parker. She's a graduate student in the Carpson Laboratory at our Florida campus. She will be giving a presentation about her research on ribosomes and their role in health and disease. And the title of her presentation is How Not to Get Lost in Translation. She will also be talking about her journey into science. Before moving into her presentation, I want to remind everybody of some housekeeping items. So all of your videos have been disabled and you have been muted. The format today will be a 10-minute presentation by Melissa, followed by a 10 to 15 Q&A session where you can ask your questions live. To do this, you're gonna use the Q&A button on the bottom of your screen. And this is the only way that you have to communicate with our panelists or with me, with our presenter today or with me, the moderator. We also want to communicate with you or interact with you, and we're gonna be doing these via polls. So I see that most of you are high school students and a few college students, that's great. And I see that most of you are here to listen to the scientist stories, to get advice about pursuing science, and to learn a science topic, which you will be doing today. And I see that you're very excited about science in general. So thank you for responding to our poll. And with that, I want to hand it over to Melissa.
Hi everyone, my name is Melissa and I'm a graduate student at Scripps Research working in Dr. Katrin Karbstein's lab. In the Karbstein lab, we work on ribosome assembly. But before I get into what that means, I just want to give you guys a little bit of background about me and how I got to graduate school. So my journey to graduate school started by growing up in the Bay Area in California outside of San Francisco. And I initially became interested in science by taking a biotechnology class that introduced me not only to science and the different kinds of experiments you can run, but also hands-on experience working with similar equipment that I work with in the lab today as a scientist. And so that just initially piqued my interest in science, and I wanted to continue studying. And so I applied to and went to American University in Washington, D.C., where I got a bachelor's of science in biology. While at American University and studying biology, I had the opportunity to work in a lab at American, studying the relationship between obesity and the progression of multiple myeloma, a cancer of your blood plasma cells. From there, I applied to and got into graduate school at Scripps Research, where I've been here for a few years, working with Dr. Katrin Karbstein, as I said, on ribosome assembly. So what are ribosomes anyways? Ribosomes are often called gene machines because they are responsible for making all the protein in all of our cells. Not only our cells, human cells, but all living cells use ribosomes to create protein. So before I describe in detail what this process looks like, we have to back up a few steps to understand what is often called the central dogma of biology. So this starts off with DNA. DNA stands for deoxyribonucleic acid. And DNA is the genetic code that contains all of the information our cells need to live and grow. Now, DNA is just the instruction manual. It doesn't actually perform the functions of the cell. And so in order to get its message across and to have cells do their job, DNA undergoes a process called transcription, where it is converted into messenger RNA. There are a variety of different kinds of RNA in the cell, but today we'll be mostly focusing on messenger RNA and how this serves as a message, as its name implies, to then be coded into proteins through a process called translation. And proteins are really, really important because they perform a lot of the functions in the cell, from just being structure to hold the cell up to actually performing functions and giving us all the traits that we have, such as hair color or eye color. This is all due to protein activity. Now, this process of converting mRNA into protein is called translation, and it is only possible because of the ribosome. Now, the ribosome, as I said, is like a little machine, and it reads the mRNA and then makes protein based on what the mRNA tells it to do. And so I found this video on YouTube that I'd like to show you about what translation looks like up close in the cell. Now, this video is going to describe the entire central dogma of biology, starting from DNA to mRNA to protein. But I just wanted to give us a quick taste about what translation will look like. So the messenger RNA is in yellow here. And the ribosome, one subunit binds first, the other half of the ribosome binds next, and it is able to then read and move across that mRNA, reading it and making protein. And we'll see in just 1 second, here we go, that there is the red protein coming out at the top of the ribosome. And so that's what it's making. So if we zoom to the end of the video, we can see that the protein is being made and pushed out of the ribosome. And as the ribosome finishes reading that mRNA, it's going to release that protein into the cell so the protein can perform its job. Now, as we got a glimpse of in this video, the ribosome is actually composed of 2 parts, or subunits, as we call them. So you have the small ribosomal subunit, also known as the 40S, in green here. And then we have the large ribosomal subunit, also known as the 60S, in purple here. Together, this full ribosome is also known as the 80S. Now, in addition to being made up of 2 different subunits, the ribosome is composed of 79 ribosomal proteins and contains 4 ribosomal RNAs, or rRNAs. Now, because it is fairly complicated with those 79 ribosomal proteins and 4 ribosomal RNAs, they have to be put together in a very orderly fashion. And this is helped by over 200 transiently binding assembly factors that help to ensure that all of the components of the ribosome are put in together. However, these assembly factors are only bound to the ribosome during its assembly process while the ribosome is being made. Once that ribosome is fully mature and ready to bind mRNA and translate, those assembly factors are no longer bound to the ribosome. Now, in healthy cells, we know that ribosomes, for the most part, are made up the same way each and every time, which is surprising because there's so many parts to the ribosome. You have all those ribosomal proteins and all those ribosomal RNAs. You think that you can make a variety of different kinds of ribosomes, but it turns out that in healthy, normal cells, you have just, for the most part, one type of ribosome that's made each and every time. And because we like to think of ribosomes as little machines, you can imagine that if a machine is built correctly, think of like this little robot or a car, if it's put together properly, it can do its job and drive. And for the ribosome, its job is to make proteins. Now, because ribosomes are responsible for making all the proteins in the cell, it is also responsible for making sure that the relative ratio of all the proteins remains consistent in the cell. a process that we call protein homeostasis. This is very important for keeping cells healthy. However, there are a group of diseases called ribosomopathies that are characterized by a haploinsufficiency of ribosomal proteins, which is just a fancy way of saying that a lot of ribosomes in the cells of these patients are oftentimes lacking a ribosomal protein, or they have mutations or something else wrong with the assembly machinery of the ribosome. So it's not built correctly. And what happens is you have a variety of different ribosomes that can accumulate in these cells. In addition, we also notice this imbalance of ribosomal proteins in cancer cells, similar to in ribosomopathy patients. And this shows us that when the ribosomes are not assembled correctly, and they don't have all of the components in the right order, that this little machine is not built correctly. Such as a car that doesn't have a steering wheel can't drive very well. The ribosome that isn't assembled correctly can no longer do its job properly of making proteins. And so this can sometimes distort the balance of proteins available in the cell. Therefore, the assembly of ribosomes and understanding the assembly of ribosomes will help us to understand more about these diseases and what helps keep healthy cells healthy. So there are a number of open questions in the ribosome field. The first of which is, how do healthy cells regulate ribosome assembly? The second is, when a ribosome is misassembled in a healthy cell, what do healthy cells do with it to get rid of this faulty ribosome? And finally, when ribosomes are not built correctly, how can they lead to disease if they're not gotten rid of? And my work in Dr. Karbstein's lab focuses a lot on this first question of how do healthy cells regulate ribosome assembly? So how are ribosomes assembled anyways? Ribosomes are constructed in an assembly line process, although a very chaotic assembly line. Ribosomes start off their life in the nucleus of eukaryotic cells, which contain the DNA. Now, that DNA is transcribed into ribosomal RNA. This ribosomal RNA won't turn into a protein, it's going to stay as RNA, and it will serve as a large structural component within the ribosome. Once turned into RNA, this RNA has to be folded and subsequently trimmed down to the correct size and bound to all of those proteins to make the ribosome. During this process, it is exported from the nucleus into the rest of the cell called the cytoplasm, where it finishes its assembly process and becomes a mature ribosome that can bind mRNA and translate. The process of ribosome assembly that the Karbstein lab is most interested in, and especially my project is interested in, is the final assembly steps of that small subunit, that 40S ribosome. During the assembly of ribosomes, we have noticed that there are a number of quality control steps that function to take the ribosome on a test drive to ensure that the ribosome is both structurally and functionally intact. What this means is that healthy cells test their ribosomes, and if they are found to be the right structure and they can function, they will be allowed to bind mRNA and translate. However, if faulty ribosomes fail quality control steps, these incorrectly built or non-functional ribosomes will be sent for degradation, which is just a fancy way of saying they'll be taken apart and either thrown in the cellular trash can, or those components will be used to build a fully functional ribosome. However, in sick cells, this process might be a little different. So not only will healthy, correctly functioning ribosomes be allowed to translate and pass quality control steps, but sometimes even those that fail quality control steps might end up skipping this degradation or garbage can step and will be accidentally let or allowed to bind mRNA and translate, even if they're not the right structure or if they're non-functional. Now, by having faulty ribosomes allowed to bind mRNA and translate, you can lead to a disruption of that protein balance that the ribosome is supposed to make, leading to more and more sick cells and ultimately disease. And so it's very important to have these quality control steps, ensuring that only functional ribosomes are allowed to participate in translation to keep healthy cells healthy. Now, in the Karpstein lab, how do we study ribosome assembly? Like I mentioned before, ribosome assembly can be very complex. And one of the many steps of ribosome assembly that I am interested in understanding more about is one of the very last steps of the assembly of that 40S, that small ribosomal subunit. And one of the final steps that we think might undergo a quality controls checkpoint is ribosomal RNA cleavage. Now, ribosomal RNA needs to be trimmed down to the correct size. So there are 4 ribosomal RNAs in a full ribosome, only one of which is in the small subunit of the ribosome, and this is called the 18S ribosomal RNA. And there are 2 other ribosomal RNAs that are part of the large subunit that I'm going to mention today, and that is the 5.8S ribosomal RNA and the 25S ribosomal RNA. Now, why I'm talking about these is because they are transcribed from DNA in one long string. However, because the small subunit and the large subunit need to be separated from one another eventually, the RNAs need to be cleaved away from each other and then subsequently cleaved to their final forms. And this involves a lot of molecular scissors to trim down these ribosomal RNAs. And so the ribosomal RNA that I'm going to talk about today that is involved in my research is in the final cleavage event of this immature 18S ribosomal RNA. This contains the ribosomal RNA 18S and then has an extra piece of RNA left on one side, as I've shown in red here. Now, in order to get rid of that extra piece of RNA so that the ribosome can then be allowed to bind mRNA and translate, you have to cut off that extra piece of RNA. And that is done by my favorite protein, the endonuclease NOB1. NOB1 acts as a pair of scissors, cuts the RNA, resulting in the mature 18S RNA. This step, the NOB1 cleaving step, is really important because at this step, once NOB1 cleaves, this RNA and it's fully mature, this ribosome is let out to bind mRNA and participate in translation. So one of the questions we have is, first, is 18S ribosomal RNA cleavage even necessary? Do cells need to do it? And if cells need to do it, is this step quality controlled? And do healthy cells ensure that all ribosomes undergo this cleavage event? And do sick cells not always make sure of this? In order to study ribosomal RNA cleavage, we like to use model organism because studying humans in a laboratory setting is not very easy. And so we like to use the model organism Saccharomyces cerevisiae, which is yeast. You probably know of yeast from either making bread yourself or smelling it baking in a bakery. Yeast is also used to make both alcoholic and non-alcoholic drinks, and is frequently used in probiotics. Saccharomyces cerevisiae, our yeast as a model organism, is really wonderful to use in lab. It grows really readily in lab very quickly. We can genetically modify these yeast to be able to understand how mutations found in diseases such as cancer or in ribosomopathies affect not only ribosomal RNA cleavage, but also how the ribosome assembles and functions overall. And It has a perk of smelling like bread in the laboratory, so it's just really wonderful to use. In addition, and most importantly, we like to use yeast because the assembly of their ribosomes follows a very similar path to ribosome assembly in humans. And so we can test out our hypotheses in yeast, and then we can eventually translate that information that we learn in yeast to humans to better understand how human disease arises. So in order to study ribosomal RNA cleavage in yeast, and eventually to be able to separate ribosomes based on their stage of development, we use a technique called polysome profiling. And polysome profiling involves taking a tube, filling it with a sucrose gradient, which is just, sucrose is sugar, so it's a very dense solution. We then break open yeast, either healthy or sick yeast, and we'll break them open and take the contents of the yeast, which includes not only RNA and protein, but also ribosomes. And we'll put that on the very top of this sucrose gradient. We will then spin the sucrose gradient really, really fast, and this will allow us to separate very dense particles from lighter particles. And so the lightest particles are any kind of free protein or free RNA, anything not bound to ribosomes. Then we're able to separate the small subunit from the large subunit and separate that full ribosome, the ADS ribosome, either just the ribosome or the ribosome bound to mRNA, here in pink. And we're able to separate that even further into polysomes. And so polysomes are when multiple ribosomes are bound to one mRNA. So in this case, we have one mRNA in pink bound to 2 ribosomes. Here we have one mRNA bound to 3 different ribosomes, and so on. We then are able to separate each of these different subunits or ribosomes and polysomes using what's called a gradient fractionator, which is this machine here. And what that results in is we are able to separate out these cellular components, and we can see that there is this gradient trace. And each of these peaks represents something. So this first peak is all of the free proteins, the free RNA, anything not bound to the ribosome. This really small peak above number 4, or fraction 4, is the small subunit. The next peak is that large subunit. Then the ADS ribosome, and all of the polysomes. What's really cool about this technique is that not only can you separate these polysomes, but you can separate individual polysomes. So fractionate here, this first peak is when 2 ribosomes are bound to mRNA. Then the next peak is 3 ribosomes bound to mRNA, 4 ribosomes, 5, 6, and so on. Now this polysome profile that I've shown you is actually from wild-type cells. Now when I say wild-type cells, I'm referring to healthy yeast cells. So we use healthy cells in our lab, but wild-type in general just means healthy, normal cells. They should contain all of the proteins and RNAs that a cell should have. These are usually used as a control for any kind of experiment. You might then have your experimental condition that has a disease mutation or is lacking a protein or an RNA that might be important to an experiment. In this case, we're interested in ribosomal maturation. So in healthy or wild-type cells, we see this polysome profile that I just described. Now in healthy wild-type cells, they contain the protein endonuclease NOB1. And NOB1 is able to cleave that immature ribosomal RNA to the mature 18S RNA. So how do we go from polysome profile to actual looking at the RNAs? Well, we take each of these fractions, so these polysome profiles have allowed us to separate where each of these numbers are individual tubes, and we can extract the ribosomal RNA from each fraction or each tube here to be able to look and see what RNA is present in the 4DS ribosome, what RNA is present in the ADS ribosome, and what RNA is present in the polysomes. And so we run out this RNA that we've extracted onto what's called an agarose gel. So I have a video for you of an agarose gel running for those of you who've never seen this before. It's really quite neat. Oh. One moment. Here we go. Okay, so in agarose gels, these are called the wells, and the wells are like little pockets inside of the gel. And you can put your sample, say in my case, RNA, into these wells. Then you can push electricity through the gel, and this will help separate the nucleic acids based on their size. So you can think of this gel as like a matrix or like an obstacle course. Smaller RNAs can get through this obstacle course more quickly, just like a smaller human could get through an obstacle course more quickly than, say, a really tall or large human. So in the same way, we're going to see these RNAs separate based on size. The smaller RNAs are going to run faster through the gel than the larger RNAs. So let's take a look. So this is a time-lapse video. In real life, this will probably take half an hour instead of just a few seconds. But ultimately, what this is showing us is that not only can you tell the relative size of these these nucleic acids. Oh, let's go to the end of the video. Not only can you tell the relative size of these nucleic acids, but you can also tell how many different nucleic acids you have in here. So here you only have one species, and it is pretty large. Here you have two species of nucleic acid. This top band is going to be bigger, and this bottom band is going to be smaller because it ran further through the gel. And same thing here. This well, you have three RNA or nucleic acids. Okay, so we extracted our RNA, we've ran out on a gel. Now we want to predict what we think we might see. So this is not the actual data. I will show you that in just a moment. This is predicted data. So once all of our RNAs are run out on the gel and we image our gel, we want to know where the RNAs are. And so I have two different RNAs here for the small ribosomal subunit. And we would expect two bands. The upper band is going to be that longer immature RNA that contains both the 18S as well as that extra piece of RNA that has not been cleaved. And then we see a band that's run further through the gel, which is shown here in black, and that represents the mature 18S RNA after it has been cleaved by knob1. So here, in this fraction that contains the small ribosomal subunit, this contains not only the mature, but also the immature ribosomal subunits. And so we would expect that we would see both this immature 18S RNA, this bigger 18S RNA piece in red, as well as the smaller 18S RNA piece, this mature piece in black. However, by the time the ribosomes are able to bind mRNA and translate in this ADS peak as well as in the polysomes, we expect that only fully mature ribosomes are able to bind and translate mRNA. And so we would expect to only see the smaller RNA piece here in black, the mature 18S RNA. So this is what our gels look like in real life. And so our prediction is correct. We see this immature ribosomal RNA is only in this 40S peak, just in the immature ribosomes. However, by the time you get to ribosomes that are bound to mRNA, you only see the mature 18S RNA, suggesting that healthy cells have a way to make sure that only mature ribosomes are translating and making protein. Next, we wanted to know what happens if cells cannot cleave the immature ribosomal RNA. So we have designed yeast cells that will lack NOB1. So over time, they have less and less NOB1 until there's no NOB1 left, at which point the immature ribosomal RNA will never be cleaved down to the mature 18S RNA. So our polysome profile looks fairly similar between the healthy wild-type cells and the sick, missing NOB1 cells. But we don't know what our gel's going to look like. We have a prediction though. So we know that in wild-type cells, the immature RNA is only in this 40S peak. So here we would expect there might be some in the 40S peak, but maybe there's even more so in the 40S peak because now very little, if no mature RNA can be made. So you have very small bands of 18S. However, alternatively, maybe cells that don't have NOB1 are allowing immature ribosomes to translate. Thus, we might see this longer piece of RNA, that immature 18S RNA, also in the translating portion of the polysomes. So when we look at our actual data, what we find is that in comparison to wild type, that again only has that immature RNA in the 40S peak, we see our second hypothesis is true, which is that without NOB1, cells are unable to keep immature ribosomes from translating. And so we see this immature RNA present not only in the 80S peak, but also out here in the polysomes, where ribosomes are translating mRNA. So if these immature ribosomes are able to participate in translation, maybe they're not bad. Maybe it doesn't matter if ribosomes are assembled correctly. However, what we see in yeast is that when we look and see how well the yeast are growing, wild type or healthy cells here on the top are able to grow really well. You see this white color. Yeast oftentimes are white on a plate. And we see that they're growing really well. They form these actually fairly large colonies for yeast all the way out here. However, cells that are lacking NOB1 are unable to grow. They start to die, and so no cells are able to grow out here. What this tells us is that although immature ribosomes can translate, they might be able to make proteins, they are unable to make the right proteins, or they're unable to support cell growth and normal healthy cell life. Therefore, what this tells us is that it is really, really important that healthy cells are able to detect and ensure that only ribosomes that are fully functional and fully assembled are participating in translation. However, if ribosomes are allowed to be misassembled and engage in translation, what happens is a disease state, either ribosomopathies or cancers, or the progression of other diseases that can lead to sick cells and unhealthy humans. Therefore, it's really, really important that there are quality control steps that regulate ribosome assembly to ensure that cells are kept healthy and humans are kept healthy as well. All right, with that, I would just like to thank Scripps for allowing me to give this presentation and for all of you guys for coming to listen today.
Thank you, Melissa, for that awesome presentation. I want to remind everybody to go and type in your questions at this moment. And while you do that, we're gonna do another poll to see how we're doing. So if you can please take a moment to answer the questions on your screen. And again, we apologize for the connection, even though we don't have control over the internet. So that's great. I see that a lot of you are enjoying the presentation about ribosomes, and they want to know a little bit more about science careers and why is studying ribosomes so important, and a little bit about your research work. So maybe you can
Sure.
answer this question while they type in their questions.
Yeah, of course. So I'll start off with why ribosomes is important. So as I talked about a little bit today, when we have ribosomes play, when they can function correctly, just like a car. If it's built correctly, just fine. But we're knowing that when ribosomes aren't built, they no longer function correctly. And so a lot of times what that means is that they might still translate, they might still make protein. But from research that I didn't have time to show you today, but we've been able to publish from our lab as well as other labs, is that what these ribosomes make is a different set of proteins. So normally you need a lot of proteins that help your cells grow and stay healthy, and you need very few proteins that deal with stressful environments. But suddenly, if you have a ribosome that isn't functioning, it might disrupt this very careful balance of proteins. Now, suddenly you don't have enough proteins for surviving stress, or maybe you have too many of those proteins. Additionally, maybe you have too many proteins to grow, and that happens a lot in cancers. What happens and why cancer is so bad is that these cells just keep dividing and dividing, and ribosomes are very, very necessary to allow for this continual growth. So it's very important to understand not only how ribosomes are built, but also how they function under different conditions, such as in cancers or during viral infection, to understand how disease states work, and hopefully to be able to either treat the patient either by directly targeting the ribosome or some other component of the proteome, so any of the proteins that the ribosome is making, in order to bring that diseased state back to a healthy state. And so I think another question was about science careers. So there's a lot of different science careers out there. There are some that require only a bachelor's degree, some that require a master's degree, and some that require a PhD. So it really just depends what you're interested in doing within science. And obviously, this is more of biology and biochemistry, but there's a lot of different realms of science, including chemistry, physics, a lot of engineering deals with science as well. And so there are jobs from working at the bench trying to do experiments. So you might be a lab scientist. You could work in academia, like at a research institute, like where I am, or at a university where you could teach science. You can also work in a pharmaceutical company where you're trying to develop drugs to help sick patients be more healthy. Outside of bench work, you could also, like I said, you could work in teaching. You can work as a lawyer, as a patent lawyer, trying to make sure that patents are secure. And different consulting companies as well also like to hire scientists because they understand a lot of how drugs are developed and how they work. And so there's a lot of different avenues you can go with a degree in science, whether it's just from a university, like a bachelor's degree, all the way up to a PhD. So it's really just what you're interested in. There's a lot of things you can do with science.
Great, thank you. So we have another question. They want to know why did you choose to study ribosomes?
Sure. So when I was an undergrad, I was studying the molecular mechanisms of how obesity leads or can further progress cancers. And so that's obviously very human disease relevant with cancer. But what we were studying was how all these cells interact with each other and how certain conditions make cancer grow faster or slower. So I became very interested in these very cellular mechanisms of how cells work, how they talk to each other, how they work under different environments, either healthy or diseased states. So when I came to graduate school, Scripps allows you to do rotations where you spend a few weeks to a few months in a lab. You get to try out different experiments and see whether that kind of science is a good fit for you. And so I did a rotation in Dr. Karlstein's lab, and I just fell in love with it immediately. I mean, what's not to like about a ribosome? It's made of protein and RNA, which is great, but I really like how we can study something that seems very basic, right? Ribosomes, they work, they make proteins. Not many people think about them, but they're vitally important because if they're working correctly, everything else can work correctly. There's proteins available for the cells to grow and to do their jobs. But suddenly, if the ribosome isn't working correctly, things go haywire in the cell. And I just became very interested in how something usually overlooked can be so important to cellular behavior and human disease.
So the next question, if you did a lot of research experiences before you decided to go to grad school, and is this something that they as students need to do to get into science?
Sure. So for me personally, I didn't have much science experience going into college. When I was in college, I really wanted to have that experience. So I did an internship in a lab for a summer during the summer break. And then when I came back to school during my sophomore year or during my junior year, I wanted to join a lab. So I talked to a few professors there about working in their labs and doing some research. And I was very lucky. I got a position at my undergrad working for a professor there. And so I had about a year and a half, 2 years of working in that lab as experience. So not all graduate students have experience coming into grad school. Most do. And that's very important for a number of reasons. The first of which is it just makes sure that you actually like science, because sometimes bench work can be a lot of work. And if you're really passionate and you're really excited about it, it can be a lot of fun. But there are some hard days to it. So you want to make sure that you like doing research before you jump into graduate school, which oftentimes is between 4 to 7 years long for a PhD. So it is something that I would recommend if you're interested in science to try and pursue research experience, even if you're maybe interested in medical school, or maybe you're thinking about graduate school or just science in general. It's good to just see how science is done on a day-to-day basis in the lab.
Great. Thank you so much, Melissa. It's already 3:45, so we're gonna go ahead and wrap up. But we really appreciate your presentation today. It was very informative, and all your questions. I want to let all the attendees know, I'm gonna start my video so they can see me. I want to let all the attendees know that we're gonna have this video on our webpage, which you're gonna find more information at the end of the webinar. We will probably record it again so we don't have the lagging issue that we have. So if you want to watch Melissa's presentation again, it will be there soon. And so right now we're gonna have another poll, the last poll, to see how we did overall. Please take a second to respond to these questions. Meanwhile, I'll tell you that the next webinar, which all of them occur on Tuesday and Thursday at the same time. So 3 p.m. in the East Coast and at noon in the West Coast. The next one is on women in science, or issues of women in science. And then we have a demo next Thursday on chromatography. So we invite you to join us for these webinars. And after we close this call, there's gonna be a survey. We ask again that you please give us your feedback. And if you want to, you have any topic that you would like us to discuss, you can enter these topics at the end of that survey. And also at the end of the survey, you can learn a little bit more about our web resources and where you can visit and get access to all the videos and the next webinars. And with that, I'd like to thank you again for joining us. I want to thank Melissa for presenting, and I'll see you next time. Thank you.